Hello and welcome to SAM online class. This is Dr. Subrat Kumar Mahanti. Today we shall discuss about small interfering RNA and microRNA. Before that, we should know what is RNA interference. RNA interference is a biological process in which RNA molecules are involved in sequence-specific suppression of gene expression by double-stranded RNA molecules through transcriptional repression or translational repression. RNA interference is a biological process which causes gene silencing due to the involvement of specific RNA molecules. The central components of RNA interference pathway are small interfering RNA and microRNA. Small interfering RNA and microRNA are the central components of the RNA interference pathway. Both of them have discovered a little over 2 decades ago. Both small interfering RNA and microRNA are non-coding RNAs that play important role in gene regulation. Both of them have recently been investigated as therapeutic agents for the treatment of various disorders including cancer and infection. Clinical trial of RNA-based drugs have already been initiated. Both small interfering RNA and microRNA are short duplex RNA molecules. Small interfering RNA target only one type of mRNA, whereas microRNA have multiple targets. Both small interfering RNA and microRNA cause gene silencing at the post-transcriptional level. Both are highly potent. They are capable to act on non-druggable targets. What is non-druggable targets? The non-druggable target may be a protein that do not have any enzyme function, or the non-druggable target may be a specific conformation of a protein that is inaccessible to the traditional drug molecules. RNA interference is also known as co-suppression, post-transcriptional gene silencing or quelling. The 2006 Nobel Prize in Physiology or Medicine was shared by Andrew Fire and Craig C. Mello for their work on RNA interference in nematode war Sinorhabditis elegans. The small interfering RNA and microRNA, they are the central components of the RNA interference pathway. Now let us discuss about the similarities and differences between small interfering RNA and microRNA. First, let us know the similarities. Both small interfering RNA and microRNA are non-coding RNA. Both of them cause gene silencing at the post-transcriptional level. So, what are the differences? The differences are small interfering RNA are exogenous RNA, whereas microRNAs are endogenous RNA. That means microRNAs are synthesized inside the nucleus itself. And these microRNAs are transcribed in the nucleus as primary microRNA. These primary microRNA molecules fold upon themselves to form hairpins. These hairpins are trimmed inside the nucleus. by a microprocessor complex. This microprocessor complex consists of two components. One component is a ribonuclease 3 that is known as DROSA, and the other component is DGCR8. This DGCR8 is a RNA binding protein. So the hairpins are trimmed inside the nucleus by a microprocessor complex to form approximately 60 to 70 nucleotide hairpin pre-RNA. This pre-RNA is transported from nucleus into the cytoplasm through nuclear pore complex. In the cytoplasm, the pre-RNA is cleaved by a protein known as dicer to form approximately 20 nucleotide microRNA. This is about the synthesis of microRNA. Regarding small interfering RNA, small interfering RNA are exogenous RNA. They can come into the cell through virus infection or through external manipulation. The small interfering RNA are also RNA duplex molecule. They are short RNA duplex molecules. These RNAs are also cleaved by the protein which is known as dicer. Then what are the other differences? One point is small interfering RNA and microRNA, both of them are RNA duplex. Small interfering RNA has 21 to 23 nucleotides with 2 nucleotides overhang at 3' end, whereas the microRNAs are also RNA duplex. They contain approximately 19 to 25 nucleotides, and this also has a 2 nucleotides overhang at 3' end. Small interfering RNA is fully complementary to the targets, whereas microRNAs are partially complementary to the target mRNA. Because of this, small interfering RNA have only one target. That means the small interfering RNA can influence a single type of mRNA molecule, whereas the micro-interfering RNA whereas the microRNA influence the expression of multiple mRNA molecules. Other differences are regarding mechanism. Small interfering RNA, they act through endonucleolytic cleavage, whereas microRNA, they cause translational repression. They also cause degradation of mRNA. And regarding the clinical application, small interfering RNA can be used as therapeutic agents, whereas microRNAs are useful as biomarkers. Friends, this is about small interfering RNA and microRNA, and you came to know what are the differences between small interfering RNA and microRNA. Please share, like and subscribe this video, and also please give your comment. See you, bye bye.